Innovation means doing the very best science possible. And one of the things I've learned being here in New Mexico is I think the most creative thinking and innovation science comes when two worlds collide and when scientists of different disciplines have to learn to talk to each other. So some of my first research projects here in New Mexico were with bioinformaticians and computer scientists and mathematicians at Sandia National Lab. And they talked in math formulas, and I talked in biology. And once we got over the sort of their disrespect that biology was too messy and could never be described in equations, and it was all descriptive, and I got over, I was a math major in college, but still got over the complex formulas and learned to talk to each other, the science was profound. And that's what my collaboration with Jeff Brinker has been like. So with Jeff's incredible and world-renowned expertise in nanotechnology, materials science, and engineering, fields that before we started working together I appreciated, but didn't have a deep understanding of, and my expertise in cancer biology, we've created something together that's very innovative. And leading the Cancer Center, it's been really clear to me that much of our very best science comes from scientists, physicians, students, community members suggesting ideas from their very different disciplines and bringing those different thoughts and backgrounds together to solve a problem. The collaboration that Jeff and I developed came about naturally. I still remember the day we met several years ago. I certainly knew of Jeff and his stellar scientific reputation, but he came into my office, or we were at a meeting together, and he kind of shrugged his shoulders and said, I'm a nanoscientist, and I've got this really interesting particle, this tiny mesoporous silica nanoparticle, and I think it really could have important applications in cancer. And I'm like, really? Thinking, that's a lot of hype, we'll see. But the more we sat and talked, I realized how beautiful his innovation really was and that it could have a profound impact in cancer research. Now, getting these experiments to a human is what we call the valley of death in cancer research and cancer medicine, because it takes millions of dollars, tens to hundreds of millions of dollars, to invest in the animal studies and the scale up of these compounds to actually do what are called first-in-human clinical trials. So Jeff and I are both really thrilled that a company called Oncotherion in Seattle have invested $27 million in this technology to try to take our studies through animal models ready for first-in-human clinical trials. We're also working on a couple other cancers. Ovarian cancer is a very good target. Ovarian cancer is often widely metastatic in the abdomen. It's why women die. And there's a protein on these cells called EGFR. So we've developed really lovely EGFR-targeted protocells, and these can be injected into the peritoneum or abdomen of women and be a localized therapy for nanotechnology. So that's a second area of investigation. We're also into animal studies. And then we're looking at applications in liver cancer, brain tumors, and other sorts of cancers where we think this technology may be amenable. So at the end of the day, what we hope we've created is a platform, if you will, where a specific protocell could be carrying a certain type of drug and have a particular targeting moiety that would make it a unique therapeutic agent for a very specific cancer. Another example that I'm very proud of in our own leukemia work is the discovery of a very novel form of leukemia here in New Mexico, which we call Philadelphia-like acute lymphoblastic leukemia. And it's been known for many years that Hispanic children, Native American children, when they get acute lymphoblastic leukemia, they have very poor survival rates on today's standard therapies. The high-dose standard therapies that children receive now for 4 years with leukemia get to survival rates of 80–85% in white children. But Hispanic and American Indian children have always had far worse survival rates, 30, 40, 50%. And it's been interesting and a bit of a front to these cultures because the presumption of our nation has been, well, Hispanic and Native American children's families are less compliant. They don't finish their therapy. It's socioeconomic status. So about 10 years ago, we began a field of research to apply next-generation sequencing and genomic technologies. And now, 10 years later, we've studied over 3,000 children with these novel scientific technologies, next-generation sequencing, deep sequencing. And we've actually discovered that Hispanic children and Native American children have very different leukemia-causing mutations than white or non-Hispanic white children. And the mutations that they have are not sensitive to today's current therapies. So importantly, with STC, we have a number of patents on these discoveries, and we've just received FDA approval for a new diagnostic that we will be screening all of the children with leukemia in the United States and the adults here at UNM, about 4,000 cases a year, to determine whether they have the mutations associated with PH-like ALL. And the really exciting thing is we found that some existing cancer drugs are very effective against these mutations. So we're going to be leading new national clinical trials testing those drugs in these children and adults. So a second project in leukemia protocells is there's a particular protein on pH-like cells called CRLF2. And so we're also working on developing CRLF2 targeted protocells to see if this might be another model of drug delivery to this very specific type of leukemia. So for me, it's a blessing, if you will, to have that science in 10 years proceed from the laboratory to national clinical trials. And we already have several what are called, a terrible word, but anecdotal cases, where as we've been doing this research, there are children who have been failing leukemia therapy. And I remember the first call about 2 years ago of a little boy in Denver who was dying. He had received induction chemotherapy for his leukemia. And the physicians in Denver had heard that we were doing the science, which we'd been presenting, because we're all part of an organization called the Children's Oncology Group that does research and clinical trials. And they said, Cheryl, do you think this child could have those mutations? So they sent samples here, and we quickly screened him, and lo and behold, he did. And they got permission from the FDA to do what are called off-label drug use, to actually take the therapy we plan to test in the clinical trials and give to this child. And today, he's disease-free 2 years later. So from death to disease-free is pretty dramatic. And we have about 40 examples of children like this now who've been treated off trial. So this fact accelerated the FDA and the National Cancer Institute approval of moving our science into national clinical trials, because the results, at least in these children already, have been really compelling. So that work is a beautiful bridge for me of science to the human already, and is also very much dovetailing into our work with the protocell. I think every cancer researcher's dream is that their science actually makes a difference in the life of a human. And for many of us, and for many science projects, that doesn't happen in our lifetime, in the span of our lifetime. We sometimes call that postdoc years. How many postdoc years does it take to take a piece of science and really get it to a diagnostic agent that could be used in a human being, a therapeutic agent that could be used in a human being? And that takes many, many, many years, and many forms of grants, many stages of research, and a lot of investment. So one of the challenges for a cancer center like ours, one of the nation's National Cancer Institute designated cancer centers, is that's the research standard we're held to. We are expected to translate our science from the laboratory or the population or the community all the way to a human being. That is a huge challenge. And one of the things we're measured on every 5 years that we go through our competitive recertification is, how well have you done that? Show us examples where your science in the lab has gone to a diagnostic or a therapeutic agent. And I think one of the things that recently really impacted our site visit team, we had 26 site visitors who were cancer center directors and great cancer scientists from all over the nation come to Albuquerque on February 23rd and 24th, and that's exactly what they were evaluating us on, among other things. But one of the points we made is, since 2010, UNM investigators associated with the Cancer Center have done 11 new startup companies here in New Mexico or across the nation from their science. And we've developed over 130 patents with STC. So patents or patents pending. So that's pretty remarkable for a center of our size. So I like to think we're already a cornerstone of Innovate Albuquerque. We've been doing this for some time. We're good at it, and it's something that can continue to grow. So the important science of the protocell is, why do it at all if it's not going to get to a human? Why do it at all? I mean, it's fun to play around in the lab, but if you're not going to make it work, then, what are we doing? So I think for both Jeff, myself, and our entire research team, getting this to the endpoint, getting across the valley of the death, bringing this technology to humans is our end goal, and we believe we can achieve that. With the new investments, with the licensing of the intellectual property, with the research collaborations we've developed around the nation, that's clearly our goal. Our grant from the Leukemia and Lymphoma Society also has us on a tight 5-year timeline. We have to, in 4 more years, be at the point where we finish these animal studies and we're ready to do the scale-up work to go to human clinical trials. And I believe we'll make it. I'm very confident.